Good morning, everyone. Good afternoon in Asia. Good morning in Europe. Good evening in Australia and New Zealand. It's good to see everybody and have you here for the first of the Angewandte Fall Symposia. My name is Theresa Kueckmann, and I'm one of the executive editors of Angewandte Chemie. I'm based in Germany. And these three events are to introduce the new Scientific Advisory Committee of Angewandte Chemie. We're holding them at different times of the day so that we can connect with audiences around the world. So this one is being held in the middle of the day for audiences in Asia. The one in October will be at a time slot that's perfect for people who are based in Europe and will be later in the day than for anyone in Asia. And then the one in December is aimed more at audiences in the Americas and will then be held at an appropriate time there. So I guess most of you already know Angewandte Chemie is a journal, but what you maybe don't know is that it's a journal of the German Chemical Society, so a society journal. German Chemical Society is also known as the GDCh, according to its German abbreviation. And the GDCh serves the chemical community in Germany and also around the world. And it plays a really important role in the development of Angewandte Chemie as a journal. To give you an idea of how that works in a little bit more detail, here's a little scheme that shows how we work together. Over here, in the bottom left, we have the editorial office and the executive committee, so the 6 executive editors of Angewandte Chemie. We're all full-time professional editors. That's our whole job. And we work together to develop Angewandte Chemie as a journal with representatives of the GDCh through the Angewandte Chemie Council, shown in the middle. And there's representatives of the GDCh are shown on the right side, include the president, the executive director, the chair of the International Advisory Board, and an additional board member from the GDCh. And then on the bottom right, we have the Scientific Advisory Committee and the International Advisory Board. These two bodies are comprised of top-notch chemistry researchers from around the world who are there to support the editorial office in different ways and to help us connect to the different voices from around the world that are important for the chemistry community. So here are the 10 advisory editors that make up the Scientific Advisory Committee for Angewandte Chemie. These 10 individuals, as I said, are there to support the editors also in the day-to-day decision-making, especially in close or difficult decisions, or those where their sort of active research expertise is particularly important. And like I said, they represent all of the different fields of chemistry and also come from around the world to help us keep those connections within the community strong. The International Advisory Board also, again, represents all different aspects of chemistry, comes from around the world from very many different backgrounds, so that the journal can continue to serve and connect with people and voices and individuals in the chemistry community as we continue to move forward. So with that, I'd like to hand over to Li-Zhu, one of my fellow executive editors, who will introduce the speakers for this session.
Okay, thanks a lot, Theresa. Do you mind staying on this so we don't have to switch? Hi, everyone. As Theresa just introduced, the three symposia for the series Fall Symposia Series features introducing our newly installed Scientific Advisory Committee, and we are able to invite all our advisory editors to present their related research with the audience, and also they will attend the meet the editor session after the presentations to communicate and exchange ideas and with the audience, with researchers. So today we are happy to have 4 of our scientific advisory editors doing talks and also attend meet the editor session after the talks. Our first speaker is Professor Li-Zhu. So Professor Wu received her bachelor's degree in chemistry from Lanzhou University in 1990, and her PhD from Institute of Photographic Chemistry, Chinese Academy of Sciences, under the supervision of Professor Chen Tong in 1995. After that, she spent 4 years at the Institute of Photographic Chemistry as an associate professor, and she also had a postdoc experience at the University of Hong Kong, working with Professor Chiming Zhe. And after that, she returned to the same institute as a full professor. In 2019, Professor Wu was elected a member of the Chinese Academy of Sciences, and her research interests are focused on photochemical conversion, including artificial photosynthesis, visible light catalysis for organic transformation, and photo-induced electron transfer, energy transfer, and chemical reactions in supramolecular systems. Next, we will invite Professor Wu to present her latest research. Because of some internet connectivity issue, Professor Wu has provided us with a recording of her presentation, but she's currently here in this meeting room, and she will be happy to answer any questions related to her talk after we play the recording. Ash, could you please?
Yes, I will.
Share the recording.
Ladies and gentlemen, I'm Li-Zhu Wu from Technical Institute in Physics and Chemistry, Chinese Academy of Sciences. First of all, I would like to thank the organizing committee for this kind invitation. It's my great pleasure to report our group's work. The title of my talk today is Artificial Photosynthesis for Chemical Transformation. Photosynthesis is the largest and the most important chemical reaction on the Earth. Photosynthesis in nature is a paradigm of the photochemical reaction which provides a blueprint for the conversion of the light to chemical energy. Fascinated by the ability of the nature photosynthetic system to convert solar energy into chemical energy, the scientific committee long ago recognized the potential of light-driven reaction as a powerful approach to chemical transformation from the high-energy intermediate generated by photo-induced excitation of the organic molecules. Unique reaction pathway can be accessed that are generally unavailable to conventional thermal reaction pathway. Our group has long been engaged into the photochemical research related to artificial photosynthesis for solar energy conversion, visible light catalysis for large-scale organic synthesis, photophysics, and photochemical properties and applications. In these presentations, I will compile the following stories to show the power of the photochemical transformation. Nature long ago figured out how to use photosynthetic complexes to capture sunlight and store its energy in a chemical form, hydrogen, in which hydrogenases can catalyze the reversible reduction of proton to hydrogen with remarkable activities. With the structural elucidation of the hydrogenases, featuring a butterfly 2-ion 2-sulfur subunit coordinated by a cysteine linked force iron 4-sulfur cluster, a carbon monoxide, and a cyano ligand, and by a disolid bridging the two ion center, scientists have been devoting considerable effort to developing artificial photosynthetic systems using hydrogenases mimic for hydrogen generation by visible light. Two different approaches have been put forward. In the first strategies, the silane bridge is employed to connect the photosynthesizer and the hydrogenases catalytic unit. The alternative way involves anchoring the photosynthesizer to one of the ion center of hydrogenases mimic. Following the two strategies, hundreds and thousands of the elegant molecular photocatalysts of photosynthesizer hydrogenases active site have been constructed. However, only a few examples can produce the hydrogen. In comparison to the efficiency of the hydrogenases mimic in nature, these systems thus far give rise to the small amount of the hydrogen and finally finish their reactions in organic solvent or a mixture of organic solvent or water. More strikingly, these synthetic hydrogenases mimics are not stable and will decompose generally within 1 hour irradiations, thus create an artificial hydrogenases systems that can produce hydrogen by visible light irradiations with high efficiency and stability. It remains basic scientific challenges. With this in mind, we introduced isocyanoligand to anchor 3 hydrophilic acetrates to the active site of hydrogenases mimics in order to enhance the solubility of the catalyst in water, and we use a quantum dot to capture sunlight and ascorbic acid as a sacrificial electron donor and a proton source. Upon light irradiations, more than 500 equivalents of the hydrogen are achieved for 10-hour irradiation, with a maximum of the turnover frequencies of 50 per active site per hour. This result implies that both the catalyst and the photosynthesized are regenerated, and the whole reaction is photocatalytic in nature. A synthetic hydrogenases mimic, even the most popular one that will decompose generally within 1 hour irradiations, could be served as an effective catalyst for hydrogen generation. This exciting result encouraged us to develop an interface-directed assembly for the hydrogen evolution. The surface affinity of the water-soluble quantum dots allow for the interaction of the sulfide atom on the hydrogenases mimics in aqueous solution. Indeed, upon a light irradiation of these self-assembled systems for 80 hours, the assemblies enable hydrogen productions with turnover number of 8,000 based on the hydrogenases mimics. Suggests this kind of the catalyst center are compared with those other systems for hydrogen generations in solution. Nevertheless, the efficiency of the hydrogen evolution is less than the nature hydrogenases, which can produce 6,000 to 9,000 molecular hydrogens per active site per second. To address this point, we introduced the first set of the polymer-based hydrogenases mimics for hydrogen generation. The system shows exceptional turnover numbers, that is 22,000, and the turnover frequencies equals to the 3.6 per second per active site. The polymer chains the assemblies actually plays the three roles in these systems. It's a framework to bring the hydrogenated active site into aqueous solution. It's a good stabilizer, protecting the quantum dot from aggregation and enhancing the emission quantum yield of the photosynthesizer. The most important thing is narrow the distance between the photosynthesizer and the hydrogenated mimics for efficient electron transfer. These three functionally jointly contributed to the efficient photocatalytic hydrogen evolution. Synthetic chemistry has allowed the preparation of remarkable close mimicry of hydrogenated mimic. However, most models of the active site represent mimics of the inorganic cofactor only, and the enzymatic like the reaction of the hydrogenated mimics is poorly understood. Actually, in nature systems, the hydrogenated mimics is deeply embedded in the proton environment. What we are interested in is it possible to develop this restricted environment to understand the electron transfer and the proton reductions in a confined medium. And here we initiate the study of tritosome confined hydrogenated mimics for hydrogen evolutions. As you can see, the self-assembled systems is quite simple, with a simple list of the hydrogenated mimic and a photosynthesizer in the presence of the tritosome can give rise to the hydrogens with turnover numbers to 52,000 turnover numbers, and the active site can produce the hydrogen with 1.4 per second per active site. The catalytic stability is enhanced from 1 hour to 60 hours, and the catalytic activities is over 4,000 fold higher than the same systems without tritosome. And this result suggests that the strong interactions and close contact between photosynthesizer and hydrogenated mimics in a confined medium is so crucial for the hydrogen evolutions from the self-assembled systems. These systems is quite like the nature systems for hydrogen evolutions that in the protein environment. Learned from nature. We are very much interested in design an artificial photocatalyst that is as abundant, stable, efficient, and sustainable. Here are the examples. We discovered a simple artificial catalyst that is only made from a photosynthesizer and as abundant cobalt salt and visible light irradiation in water. Upon irradiations, the generated photocatalyst ensued is able to produce the hydrogen from water with turnover numbers of 220,000 at room temperatures. And this kind of the catalyst is quite stable and can be reused many times without losing the activities. To understand the generality of the artificial photocatalyst, we made a simple systems only from the cadmium selenide quantum dots and nickel salt with visible light irradiation and at room temperature. This artificial photocatalyst is able to produce hydrogens with a much higher efficiency and stability. This result greatly enriched our understanding for making the cheap, robust photocatalyst using the quantum dots and the metal salt for the hydrogen production. The most important things we found upon light irradiations, the catalyst formed ensued in the reaction systems. These examples show you that upon light radiation of the quantum dots and the metal salt, there's a hollow structure photocatalyst formed immediately in the reaction systems. In comparison to well-developed traditional approaches, the hollow structures are formed in suit by the photo-generated hydrogen bubbles at room temperatures. The average diameter and shell thickness of the nanosphere ranged from the 10 to 20 nanometers and from the 3 to 6 nanometers, which are rarely reported in the literature. And the most important thing is the hollow structure shows the excellent photocatalytic activities and stabilities for the generation of the hydrogen. Compared with the biological systems and the biomimetic systems for the hydrogen evolution from water, the discovery of the artificial photocatalyst established a new paradigms for creating the photocatalysts that are highly active, robust, cost-effective in aqueous media. In nature photosynthetic systems, the light-absorbing chromophores are well organized to form the lightest harvesting assemblies and deliver the photo-induced energy and the electron to the reactive centers. This is very important for conversion of the light into the chemical energies. To duplicate the nature systems, we developed systems that is made photosensitizer surrounding the catalyst centers. And what we are thinking about, we could collect the light energies by the chromophores and immediately to transfer the multiple electrons to the active site for the hydrogen generations. As you can see, upon light radiations, what we achieved, the turnover numbers equals to 16,000 million turnover numbers for the hydrogen evolutions. You can see this kind of the systems is quite stable, and the systems can give me an initial internal quantum yield of the 65%. The abilities to assembling of the nanometer size quantum dots and the catalyst, tunable and friendly functions, just like nature does. And from the very beginnings, there's no examples can conduct such activities. Over past several years, as you can see, the stabilities and activities of the artificial photosynthetic systems is enhanced dramatically, even can approach to the practical applications. Take home message, that is, upon light irradiation of the photosensitizer, the photo-induced electron transfer takes place. The larger driving force for the electron transfer, the large number of the electron transfer rate will be. Following the electron transfer, the reduced active sites can interact with the protons in solution, and in this case, the longer lifetime of the reduced intermediate, the large amount of the hydrogen will generated from the reaction systems. Is it true that is the photo-induced electron transfer to the active site? And the active site, reduced active site, interacted with the protons for the hydrogen evolutions. How to know that? We use the time-resolved transient absorptions to monitor the photo-excited photosensitizers pathways. As you can see, upon light radiations, the photo-induced electron transfer takes place so fast, and the times is in the 65 picosecond range. That means the photosensitizer deliver electrons to active site. On the other hand, is it true the active site accepts the electrons from the photosynthesizer? And in this case, we provide the first examples that using the time-resolved absorption spectrum to give the picture of the reduced active site spectroscopic signal. And the time scale for this reduced signal is in the 60 picosecond. That means the photo-induced electron transfer from the photosynthesizer to active site takes place. Here arises a question. 2 electrons are required for 1 molecule of hydrogen. After the first electron transfer, what is the next for the reduced intermediate? To understand the following transformations, we designed a hydrogenesis mimics. During the hydrogen evolution, what we found is a new species in the course of the reaction. And the experiment and the DFT calculation demonstrated this new species is a dimer structures. And the isolated this dimer shows a real catalyst roles for the hydrogen generations, while the mimic one worked as a pre-catalyst and involved the hydrogens only after being reduced to 2. The remarkable activities and the catalytic reactions of this dimer structure of 2 operated in the photocatalysis. On the other hand, semiconductor quantum dots in conjunction with the Earth-abundant metal ion to generate an ensued catalyst is so effective. The exactly in structure changes of this active site at realistic conditions stimulate us very interested to give these details. While in this way, we successfully track the structures, the variation of the catalyst site in the typical hydrogen generations. And the nickel precursors changes to the nickel hydroxide nanogate in an alkalic medium. Further, operando the spectroscopic technicals, APR shows that actually the photo-induced transformations to give a new structures of the catalyst, that is the nickel vacancies and the nickel cluster. DFT calculations shows actually the water molecules interacted with the nickel vacancies and the protons interacted with the nickel clusters in this species for the hydrogen evolutions. Actually, the nickel clusters behave like hydrogen evolution site, thereby promoting the activities hydrogen's photogenerations in the alkaline medium. Well, to understand the whole process, can we make a real artificial catalyst for the hydrogen generations? This is examples. We just make a very small nanocrystals, integrated catalyst, and the protectant layer on the surface, a 2-step strategies for the site and the spatial selective integrations of the abundant element into the catalyst. Well, by taking the anisotropic zinc sulfide shell as a medium to anchor the catalyst, the synthetic catalyst enabled to capture sunlight and induce electrons to the active site and the catalytic center at the active site for the hydrogen generations. These strategies provide a real catalyst for preparing the multifunctions of the ultra small artificial photocatalyst for the hydrogen evolution. By mimicking the hydrogenesis in nature, we have established the artificial hydrogenesis to reduce the proton to hydrogen with remarkable activities and stabilities. At the same time, we also mimic the oxygen evolution catalyst in nature to deliver oxygen from water. And in this case, we construct this integrated systems to split water into oxygen, and the proton and the electrons delivered to the other active site for the hydrogen evolutions. Following this kind of the pictures, we have achieved the water splitting into hydrogen and oxygen. At the same time, use the carbon dioxide as a substrate in water systems to reduce this very challenged substrate into value-added chemicals. From these studies, what we think about that by integrated the photocatalyst, that is for the light harvesting, charge transfers, and the exciting utilizations into a one systems, that is very helpful to realize the very challenging chemical reactions. Finally, I would like to thank now groups that the supermolecular photochemistry group in China, and I would like to thank the National Science Foundation of China, Ministry of Science and Technologies of China, and the Chinese Academy of Sciences for financial support. Thanks for all your kind attentions.
Great. Thanks a lot, Li-Zhu, for your presentation on using photochemistry for various chemical transformations. It looks like we've got a couple of questions from the audience, and I've sent the first three to you using the direct message. So the first ones are from Dr. Arain. I think he wonders the cost of artificial photosynthesis in general, I guess, and also he asks, can artificial synthesis turn back climate change? I guess whether artificial photosynthesis would offer alternative for maybe saving some of the high energy costing transformations in industry things. And later, he also asked questions whether there are any risks of artificial synthesis systems for chemical transformations. I will also pass the question to you using chat.
Okay, I try my best to answer these questions. For the cost of the artificial photosynthesis, I think it's very dependent on the what kind of the catalyst you used for the chemical reactions. In our system, as I showed in our presentations, I think the cost is the best performance is around $2 to $3. Nowadays, we evaluated this kind of the catalyst, but that's very cheap for this kind of the reactions. For another questions, that is for the climate change. I think this is a long goal for this kind of the studies. And nowadays, a lot of the catalysts have been developed for carbon dioxide reductions, and I think the performance is so good. But for convert this kind of the stable chemicals to value the chemicals, I think there's still need some time to go. And from this point, I think that the nature systems, that is kind of the carbon balance systems. So I think if we can develop the very efficient systems for this kind of the transformations, we can go like the nature to convert the carbon dioxide to some clean and to make this climate change something change. And another thing is, I think for the climate change, that is the industry development to make this carbon neutral, carbon rich society. And in other ways, we must develop some of the systems that very efficient to convert this kind of system to neutralize this kind of the situation. That's my understanding. Xing, is this okay?
Thanks a lot. Thanks a lot, Li-Zhu. Another question from Kamini. Sorry, I couldn't see the full name here. So he asked, we use lanthanide ion doped titanium oxide for catalysts capable of upconverting infrared radiation to visible radiation to capture part of the 50 to 55% IR portion, in addition to 5% UV and 40 to 45% visible for water photosplitting. We get continuous hydrogen evolution when a sacrificial whole scavenger, such as ethanol, is used in water. And wonder whether you have materials for up-converting infrared to visible.
Yes, what we are doing is quite different from yours. You just use lanthanide ions to dock to the TiO2. That kind of the system is interesting. But from our side, we developed some organic compound that can absorb the light very efficiently, and then converted the long wavelength energy to the short ones. That's I think the principle is the same, but the way is different. And now what we found is this kind of the system is very interesting and can enhance the light harvesting performance for the catalyst. Yes, thank you.
Great, thanks, Li-Zhu. And one last question is from Zita. I have a question that hydrogenase is a reversible catalyst for one-way activity to make hydrogen efficiently. What pH will be more suitable for hydrogenase?
Yes, this is a quite good question. And that is, it seems that the acid medium is good for the hydrogenases, but in nature systems, actually it's a neutral system. This is very dependent on the electron transfer and the proton transfer. In this case, if we can cooperate the electron transfer and the proton transfer immediately, we can keep this kind of the reactions even in the media and basic solutions. We have already achieved this kind of the system very efficiently, even compete with that in the acid solutions. So pH is important, but not only one factor. The key is the electron transfer and the proton transfer to cooperate with each other for this efficient transformation.
Okay, thanks a lot, Li-Zhu.
Okay, thank
now,
you.
It's already 2:40, so we should move on to the next presentation. Thanks a lot again, Li-Zhu.
Thank you, Xing.
Share. Slides. Okay, I'm just turning back to the slide. Our next speaker is Professor Hiroaki Suga from University of Tokyo in Japan. And Professor Suga is currently in the Department of Chemistry Grad School of Science in University of Tokyo. He received a PhD at MIT in 1994, and he did his postdoc at Mass General till 1997. Then he joined State University of New York first as an associate professor and then tenured associate professor. And then he moved back to Japan as a professor at the Research Center for Advanced Science and Technology at University of Tokyo. and he's been at his current position since 2010. He's recipient of many awards, including most recently the Research Award of the Alexander von Humboldt Foundation 2020. And today, he is going to show us the next generation of random non-standard peptide integrated discovery system. It's all yours, Hiro.
Okay, thank you. Can I share my screen? Thank you.
Yeah, please.
Yes. Okay, I hope you can see this screen. All right, I'm going to start my presentation. It will be a completely different the topic that which you have here and the previous speaker. The next generation of the rapid system, which I'm going to explain about what the rapid system is. By the way, I'm the medicinal chemistry, bioorganic chemistry, chemical biology kind of field. So the talk is related to that field. Okay, so this is the first slide that I show you. This is the kind of current drug development. The molecules that usually the pharmaceutical companies are interested in, and these are the molecules mostly on the market. The small molecules, the molecular weight is about 500 Dalton, and then more recently, a big boom of the biologics, including antibodies, which is about molecular weight is 150,000 Dalton. So there are a huge gap between the 500 and 150,000 Dalton, which is there's a pros and cons in a small molecules that can get into the cells. On the other hand, antibodies cannot get in the cells at this moment. And also specificity-wise, antibodies are so great compared to the small molecules. So I have been interested in devising a new modality, which is a different kind of the molecules that which makes the benefits of the two good part of the molecule. So I focused on the last probably nearly 20 years of the macrocyclic peptides. This is we're mimicking sort of naturally occurring macrocyclic peptides represented by, for instance, the cyclosporin A, which is the immunosuppressive agent isolated from fungus. The reason we are interested in macrocyclic peptides is because this is a chirality rich and then three-dimensional rich and conformation rich. So there are so many variety of the possible conformation of the three-dimensional structures can be realized by this particular molecule. And then molecular weight is still very low side. It's 1,000 to the 2,500 Dalton. So unfortunately, there are not many great methods to isolate or define the molecules that which is biologically active from the artificial library. It's still before what my technology has developed. Most people rely on the isolation from the natural as a natural product. So I decided to develop the three technology. First technology is the FlexZyme, which is the flexible tRNA acylation ribozyme. This enables us to rewrite the genetic code. And when you write the genetic code newly containing a certain amino acids that which you have seen in a natural product, and then you can use the in vitro translation system, which we call the FIT system, flexible in vitro translation system, allows us to design a genetic code and express peptides as you wish. And then furthermore, we couple together with the display system, it's so-called mRNA display, with this FIT system, and the FlexZyme enables us to screen very quickly random non-standard peptides. And so we call it the rapid system. As I said, it's a very fast technology to discover desired macrocyclic peptides. So I'm going to get in touch of these technology briefly because I don't have enough time to get into the details. So please read the papers that which we published. you are interested in. So this is a Flexzyme. This is a 45 nucleotide or 46 nucleotide of the RNA molecule. This RNA is a catalyst to catalyze the amino acylation on the transfer RNA, which is playing an important role to write the genetic code. So this RNA will bind to the 3' end of the transfer RNA, and it binds to the like this. This is actually the X-ray structure we solved and published in Nature quite some time ago. And active site allows us to bring amino acid in a close proximity of the 3' hydroxyl group of the transfer RNA. So the beauty of this Flexzyme is going to recognize any kind of amino acids. So the Flexzyme basically binds to any type of the tRNA, but also binds to any type of the amino acid. As far as this has the dinitrobenzyl ester or chlorobenzyl thioester, we have a few different kinds of the Flexzymes that enables to distinguish different kinds of the leaving group. It's importantly, the side chain was not recognized or amino group was not recognized. Therefore, you can change the side chain as whatever you like, but you can also press the modify the amino group, like N-methyl group or N-acyl group as you like. You can flip the chirality from L to D amino acids. And even you can do the beta amino acid. We recently published a few papers out of this technology. So beauty of this is that we are not now strictly controlled by alpha amino acids, and particularly alpha amino acids that nature uses in the ribosome translation system. We can actually redesign the translation genetic code and translate the peptide containing these amino acids. And for instance, that this is the initiation table on the right with the elongation table and the forming that after the formula methionine and translates the elongating and the different amino acids based on the messenger RNA sequence. As you probably know, these days we have a vaccine of the messenger RNA, and this is actually translated inside of our body, but this is all in vitro E. coli system that we run. So from here, we remove the methionine. The FIT system enables to make a deficient amino acid translation system. And then we are able to replace with this unique amino acid by the Flexzyme to charge to the initiator tRNA. So this case is a chloroacetyl D-tryptophan. As an example, we can do anything we want, but this is one of our particular interest of the amino acid. And then following that, we use a random sequence. We use in the case of this examples, we use NNU, and so genetic code will enables us to read all these black letters amino acids. And then furthermore, we remove the certain amino acid to replace something unique amino acid. Let's say, for example, we remove these 4 amino acids, and then we replace with N-methylphenylalanine, N-methylserine, N-methylglycine, N-methylalanine, which try to mimic the structure of the cyclosporine A containing multiple N-methyl amino acids in the backbone. So when design a random sequence, we start from initiator, which is chloroacetyl D-tryptophan, and then the translation system will synthesize, we hijack the ribosome to synthesize this peptide containing N-methyl amino acids and randomly occurring in the random sequence that we could generate from the messenger RNA. And at the last, we add the cysteine. So the length of here is about less than 15 residues containing a variety of different lengths. But it's because we have a mixture of these random sequences, we don't know exactly what we are making it, but we have a huge number of the diversity. I will get into that. So when we express the peptide, we have some linker peptide we designed, and then cysteine residues spontaneously react with N-terminus of the chloroacetyl group, which we brought from the initiator amino acid. And this reaction takes place and it spontaneously makes a macrocyclic peptide by the formation of the thioether bond. Thioether bond is a very stable bond in vivo. And so we are able to get really the nice macrocycle. The diversity we have here is a 10 to the 12 different sequences. So 1 trillion different sequences comes from the dsDNA random sequence of the DNA template, go through all of these, and we have 1 trillion different sequences. Of course, we cannot screen individual trillion sequences because this is too many to look for the active molecule. We're working on a single molecules basically, but so in order to look at it, we attach the messenger RNA. I'm sorry that I don't have enough time to get into the how we are going to these two attachment occurs by the ribosome. Please read our papers, but this is an excellent method of connecting the encoding sequence of the messenger RNA to the peptide. So we know that what the peptide sequences are, and after that we're going through the Darwinian selection type of the approach to isolate the most active sequence, the binding to the target of the choice, we get we can actually read this sequence by second generation sequencer these days. So this is a summary of the technology. I'm not getting into the details, but as I said, 1 trillion different sequences can be screened very rapidly. It's about 2 to 3 weeks to complete the selection, and the extremely cost performance is great because it's only we use 100 microliter translation system. Later on, we use only 5 microliters, so it's really almost nothing for the cost of the waste. And so it's very friendly for the environment as well. And then finally, when we get the desired molecule, we can actually do the chemical synthesis. Of course, this part, if its activity is great, we don't need to use a huge amount of the molecules. And in fact, we usually get really high active and high specific molecule. So this is the most recent paper we published in Nature Chemistry that are using a new genetic code containing a cyclic beta amino acid. These amino acids are notoriously before that we are unable to put into the library, but after the many experiments, thanking to the efforts by the Dr. Kato, made a new tRNA enables us to place these molecules into the genetic code, and then we tested for the peptide selection against one of the drug target. It's called the factor 12A, protease. And then we find the molecules that which is very strongly binds to the target, like picomolar range. This is the 1 nanomolar picomolar range, and then this is also the low nanomolar range. And also these are strongly inhibit the target protein enzymatic activity. And we have looked at how stable this peptide. Generally, that the peptides are very rapidly degraded into the serum. On the other hand, the peptide we generated this method is over 10 days, it's intact. So it's a really stable peptide can be generated. So we also solved the X-ray structure. And you see here the beta cyclic amino acids actually bind to use the molecules to interact with the protein. And we have another beta cyclic amino acids in here, which also interact with the protein surface. And then, of course, the active site is covered by the residues, is arginine that completely inhibit this enzyme. I switch another gear to explain the experiment that we actually have done. Is this a target for the plexin B1? I'm not going to get into the details about this target, but this is a pretty important role of bone homeostasis. Our body always creating the bones and then destroy the bones, and then we have to keep the disbalance to make it our bone too strong. But certain diseases, we lose this the bone homeostasis, particularly in this case is a semaphore D protein interact with the plexin B1 and it stops the differentiation of the bones, so you cannot make a new bones. So we tried to find the inhibitors of this protein interaction. Then we used our technology that we have isolated this KD is about 3.5 nanomolar. It's very strong binders for the plexin B1, and this molecule turned out to be allosterically inhibiting the interaction between the plexin B1 and semaphore D. Then we collaborate with the bone homeostasis scientist and making the testing whether we can actually get the bone homeostasis to control. And here we show you that the mouse cells that it's called the osteoblast cells, and control experiment, we add the semaphore D, you see the less number of the red count of the cells. So that means inhibited, we add our macrocyclic peptide, we can strongly induce differentiation of the osteoblast cells. So we decided to take animal studies. To do that, we actually generated this particular interesting dimeric peptide. And then we use the animal models that which doesn't have the overing in this field, that when you remove the overing by surgery, the female mice gets really weak bone. And so we administrate our macrocyclic peptide 4 times over the 13 weeks. There are lots of data here, but I show you the bones. It's very clear. So this is a wild type normal mouse. And then when you remove the overing, you see that the bones inside is very weak. You can see that less number of the clustering the bone. And then we administrate our macrocyclic peptide. As you see here, the dose that you see is even probably stronger than the wild type. It's really make a nice, strong bone can be generated. So we're very excited that we can move forward this type of the drug development. The last topic in the collaboration with the Shokhat group in the UCSF, targeting to KRAS G12D mutation. This is a reason that this is the most difficult target. So far, there is no drug available for this inhibitor. And also it's very important that we wanted to find something, a new challenge to look for the inhibitors for GTP bound form selective inhibitor. So we set the problem of the very high bar and we try to find our macrocyclic peptide against this idea. And I'm not getting details of how we did this, but we just simply use our technology to isolate only the macrocyclic peptide selectively binds the GTP form, not the GDP bound form. The reason we want to have the GTP bound form is we can completely knock down the wild type KRAS activity by doing so. Then you can expect that we don't have a side effect by this type of the drug. So we've succeeded at isolating these 2 types of the peptide. We total of the 3 peptides, but 2 are the most active, and then they are equally active to inhibit selectively G12D over wild type KRAS. And also these 2 molecules are very selective to GTP bound form. So in the past, people have isolated the inhibitor, but they are not the GTP bound form selective, therefore there are a possibility for the side effect. But here that we are completely selective to the GTP bound form. So the power of the 10 to 12 different sequences of the macrocyclic peptide and use the high pressure of the selectivity selection, and then we are able to isolate that. We got the X-ray structure, and we are very interested. We found the very interesting the structure movement. This is a very fine structure of the macrocycle. When it binds, actually the target protein has changed the structure. This probably directly connecting to the inhibitions of the protein-protein interaction. And this is a movie for that. When the peptide binds the target, it's actually changed the structure. And then this is GTP binding to the active site. So we never be able to expect it that the drug can drastically change the target structure so well. The bad news was actually this was not the membrane permeable. We're still working on the membrane, improving the membrane permeability. So this is one of the issues that we faced. We have very great technology to make it the N-methylpeptide macrocycle. However, we often suffer the membrane permeability. So my group is still working on finding a new technology and devising a new technology to solve this problem. And we always goes back natural product. Natural product is somehow a billion years evolution produces this membrane permeable macrocyclic peptide. So this is one of them. This is actually lactazole A, which doesn't have any activity, but it's membrane permeable. Most of this analog of this is called thiopeptides are often the antibiotic activity. But we are interested in this type of the molecule containing different structures. So what is the difference between them? So I'm sorry, they're going back in one slide. The structure showing here is like a pyridines closing the ring, and also this heterocycle and the dihydroalanine existing. So these are probably the structure features makes it these molecules to be membrane permeable. So there is enzymes involved, and then it's impossible for us to put these molecules into the genetic code. So we needed to use the enzymes to modify the backbone of the peptide. So people have already isolated this type of the peptides and studied it. And so we learned these things, and then we decided to work on this lactazole synthase. And two very important individuals involved in the Dr. Goto and Dr. Vinogradov, and in a collaboration with Onaka's group, the who actually first isolated this lactazole A. So this is a very complex enzymatic biosynthesis. We wanted to do at the end is this complex natural product is synthesized in one tube in one pot. So we decided to study individual step. So first we use our translation system to synthesize the peptides. And then there's an extra sequence of it's called leader peptides. And then there's a coarse peptide sequence that will be modified by enzyme. And this leader sequence is important to recruit the enzyme effectively to modify this part, core peptide region. And then we have studied the lactazole D, E, it's called LAS-D, LAS-E, LAS-F, that which makes it this azoles, and the LAS-B, and the combination of the LAS-F actually makes a dihydroalanine. We isolate these enzymes, and we studied it, and then we confirmed that they are actually active, and then they can modify them. And then the last step is actually making a 2+4 cyclization of the one of the tautomerized the dihydroalanine with couple, and at the same time it cleaves this leader sequence, the extra sequence that present in the precursor peptide, and then you form the macrocycle closing by pyridine ring, and then you form eventually the lactazole structure. So we studied all these enzyme individual steps, and then finally we can make a mixture, and we can make a one-pot synthesis of the desired lactazole A. Although we have a sort of two steps, but everything is a one pot. So once we get to this system to work in vitro, we can actually change the sequence as you wish. We're actually doing a scanning of the all A, and whatever you see here, the peak, they're formed the desired macrocycle. And you see here, most of the cases we can see the peaks except a few spots that which we cannot touch because these are important for macrocyclization. So we have only these total of the 5 residues that are critical. Well, actually, it's less than 5. It's actually 4 residues are critical for that, and we published in Nature Communication about this work. And once we know what was the requirement, and we can change the sequence and also the ring sizes. You see all these ring sizes except the first one. We see all the peaks, and so eventually we get the variety of different sequences with the different ring sizes. And this is an example. Smallest size is like this. Largest size we tested is this. And then this is the same size as what's a natural product, but we can add extra sequence of whatever you would like. Once we have this, and then we can combine together with the genetic code reprogramming, you can place different kind of the unique amino acids, and then you can still get enzyme to work to macrocyclize, and then you can get to the, now this is a pseudo-natural peptides, lactazole A, that which never exists in nature, but we can create it in vitro. Now we are interested in this is a display system. This is just we published, and that display is a possible ways to show this system. I'm not going to go get into details, but I show you here that we can scan the individual sequence by all 20 amino acids, and we see exactly the same kind of trend as before. And they can actually randomize the sequence in the middle. So we can now generate and display. So you can generate completely artificial natural product, pseudo-natural product, and display format. So you can screen rapidly, as what we demonstrated in macrocyclic peptide. So we already have done display, and the selection was successfully done. And then we find recently that the molecules that we found was membrane permeable. So these results are coming up. So it's a new kind of the generation of the rapid system that allows us to screen synthesizing unnatural products, we call pseudo-natural products, and display in the library of the 10 to the 12 different sequences, and you can choose any target you want. In fact, we actually chose the one enzyme from the human. We can completely inhibit it. So I'm thanking to all people here and my group, and very the people comes from wide variety of the countries, and thanking to my funding agencies, and thank you for your attention.
Thanks a lot, Hiro, for the very nice presentation. It seems we actually don't have any open question for now. Just one last chance for the audience to raise any question you may have to Hiro, and then okay, it looks like you did a really, really good job explaining the rapid system.
Thank you.
Here we go. Okay, so we've got some time for a question, and we do have one here. So I think the question from Barrett is about the efficiency of the system you introduced. And
so yes, the system that the peptide, macrocyclic peptide synthesis takes an hour, and we can display them, we can screen them, and we can finish the one round of the selection in one day. And then we can finish the whole entire process to discover the peptide is probably 2 weeks, including sequencing. So it's extremely fast. As a drug discovery, it takes usually a year or something like that. And then the last topic of the next generation of the pseudo-natural products, it's a little longer because it's a translation, and then we have to incubate with the peptide with the enzymes. So it's about 24 hours. It's the next day we can actually do the selection, and then you can do the round. So it's a double of the time of the selection process, but I think it's total of something like a month or less, like 3 weeks instead of 2 weeks. So it's a very fast the screening process, and then the synthesis is very efficient, as you can imagine. You have to unmute.
Oh, sorry. Thanks a lot, and thanks for the reminder. Okay, thanks again for your presentation and your answer. In the interest of time, we're actually right on time to move on to the next one.
Thank you.
And I will share the slide again. So, oops. Here. Alrighty. So, our next speaker is Professor Martina Stenzel from Australia. So Professor Stenzel studied chemistry at University of Bayreuth in Germany. And she completed her PhD in 1999 at the Institute of Applied Macromolecular Chemistry, University of Stuttgart. And after her PhD, she moved to the other hemisphere to University of New South Wales in Sydney, Australia. First as a postdoctoral fellow, and then she stayed on as a lecturer, and she was promoted to full professor in 2012. Martina's interest includes the design of new polymeric materials for biomedical applications with a focus on polymers for drug delivery. And today, she will introduce us to Glycopolymers and opens the world of squid polymers. I will now hand it over to you, Martina.
Okay, thank you very much. I just want to share my slides, please. So it's a real pleasure to be here and in this very versatile symposium. So I learned a lot now about peptides and photosynthesis. So I again would like to change gears a little bit and talk about polymer science and basically about making sweet polymers. So let me just okay, so I'm very interested in combining nature's building blocks and synthetic building blocks, such as polymers, to really get the best out of both worlds. And the area I'm particularly interested in is glycopolymers. So what's the inspiration for that? Carbohydrates are very abundant in your body. So they can be energy source. Of course, we all like sugar and we like starch food, but it's very important to understand that sugars are used to, for example, regulate cell processes. Cells communicate with each other using sugar receptors on the surface. And so when we look at the cell membrane like that, there's really a lot going on there. And there, for example, glycoproteins, there are glycolipids. And this is how the cell communicates. And the molecules that like binding to sugars, they're called lectins. And they're called lectins because they're quite selective towards very specific sugars. And we have a whole range of lectins available in that. For example, very simple lectins, which we find in our plant world. But there are also mosaic lectins. These ones can be very, very diverse. It depends very much where we find them. For example, we can find them in the influenza virus, or we have here the isoleucoglycoprotein receptor, which is, for example, very specific to galactose. And these lectins are, in that case, anchored to the cell membrane. And we have here the head and the stalk, and different parts have a selectivity to certain sugars. Then we have also macromolecular assembly lectins. These are the lectins we predominantly find on bacteria. So what is now very interesting about lectins is that they bind very strongly to the ligand, in our case, to sugars, but the binding is actually not very strong. So if we think of a lectin, they might have, for example, 2, 3, 4 binding sites here, and they might react with a sugar, but this ligand receptor complex is a very, very weak complex. So what nature does and what we can simulate as well is that instead of having single sugars, we combine these sugars together into a little complex, and now we get a much stronger bond. And this is called the multivalent effect. So the multivalent effect has been now shown with a lot of different molecules. So we can start off with very simple sugar-based molecules, which we call the glycocluster, which might have only 2, 3 sugars attached. We can now go a little bit up in size to dendrimers or glycopolymers, which might be in the range between 5 and 10 nanometers, or we can make really big glyconanoparticles. And all these molecules have in common is that they have this simultaneous binding to lectins on the surface of the cell membrane. The types of molecules I would like to talk about are glycopolymers. So glycopolymers are sugar-based polymers. So they're synthetic polymers, and they have pendant sugars. And this type of sugar can be very versatile. And so here I'm just showing some very simple sugars, but of course, we know that disaccharides, for example, or much more complicated sugars, which can be attached to polymer chains as well. And so we have here a repeating unit, meaning that we can here have up to 10,000 repeating units with the same sugar unit. So there are a whole range of different ways to get to so-called glycopolymers, and I don't really want to bore you with too much polymer science. I just want to show you here the versatility, how we can make glycopolymers. And it's always the same, really, that we have a polymerizable unit with a sugar attached. Very often we protect our sugar, our OH groups here, and we can carry out ring opening polymerization, romp or radical polymerization. And with all these kind of techniques, we can create now a whole range of polymer architectures. So this could be simple linear glycopolymers. We can also have block copolymers. A block copolymer means that we have a polymer chain with two different properties. So one property here, the red one, has sugars attached, and the other block might be very different. It might be a polymer that's hydrophobic, oil soluble. And architectures like that can self-assemble in the micelles. Of course, we can now also take our polymers and graft them onto other things. So we can graft them, for example, onto gold nanoparticles, or we can graft them onto graphene. The limitation is really just your imagination. So using glycopolymers to study lectin interaction is not just of value from a theoretical point of view, but there's some real application. And the picture here is not our own work, it's work from Rainer Haag. And he demonstrates here very nicely this principle, how glycopolymers can, for example, inhibit a virus invasion. So in the very first picture, we have here a virus, and this virus now docks onto the surface of a cell and would like to basically enter the cell membrane. This interaction between the virus and the surface of the cell is happening now via lectin sugar interactions. So what we can do now is we can add free sugar into the solution. And by adding free sugar, we basically occupy now these places here. That works reasonably well, but it's not terribly efficient. So we have this free sugar that might go in and out of these little cups here, but it's not enough to stop the virus from attaching to the cell surface. What really works, however, is having glycopolymer where we have multiple copies of this sugar interacting with that virus. And now you have a few sugars here that might not engage with the virus, but of course it's never far away, so it's not diffusing off, it's very close by, and it might interact again with the virus. So this is the concept of multivalency. So the question is, can we use glyconanoparticles to treat diseases? So obviously we can inhibit the invasion of viruses, but the area I'm very interested in is using glyconanoparticles to deliver drugs. So the typical drug administration suffers from very systemic distribution of the drug in the body. So we inject our drug, and the drug goes pretty much everywhere. It very often does not reach the target where we want it to be. And as a consequence, we have side effects. And very often the drugs have very low circulation time. So the drug might cleave very early on, might be eliminated from the body. So using now a vehicle to improve the delivery of drugs is a very old concept already. And it was first coined by Paul Ehrlich, who proposed something called a magic bullet. So that magic bullet has a pharmaceutical carrier And this is nowadays our nanoparticles. It can have several drugs inside, but it also has targeting ligands. And these targeting ligands now help us to deliver the drugs to a very specific target. And in our case, these targeting ligands are our sugars. So we would like to use some lectins that are located on certain areas in the body and target them with glycopolymers. And so the idea is now that our drug does not circulate around in the body anymore, but the drugs are now encapsulated in nanoparticle and accumulate in the specific site where we would like it to be. And so sometimes there could be a passive targeting, but what we would like to achieve is active targeting to this site of action. So once these nanoparticles reach the surface of, for example, a cancer cells, these nanoparticles are taken up into the cancer cells. And here's an example using isoglycoprotein receptor. And these receptors, for example, specifically respond to galactose. And the idea is once these nanoparticles interact with these lectins, we get very good binding, and we get now an excellent uptake by the cancer cell. So, however, one problem is, of course, lectins are basically protein structures. And of course, our body has an abundance of proteins. There might be blood proteins, they're everywhere. And so we need to now consider what happens really with our nanoparticles. Can they really specifically interact with the surface receptor, or might they interact with blood proteins? So I will come back to that one a little bit later. So an example what I would like to show you is the GLUT5 transporter. So the GLUT5 transporter is very specific to fructose, and it's interestingly overexpressed in breast cancer cells and tissues, but you cannot really find it in healthy mammary tissues. So there's already this idea if we can potentially target GLUT5 transporters using fructose. So the first step would be that we make our glycopolymers using a process called raft polymerization. It's a very easy way to get a whole range of so-called block copolymers. And in these block copolymers, we can now easily change how long we would like that hydrophobic block to be and how long we would like that glycopolymer block to be. So the length of the hydrophobic block very much influences how big our micelle is going to be. So this is actually now a very nice tool to get nanoparticles of different size and different shape. So by changing that hydrophobic block here, we might start off with very small micelles. We make that hydrophobic block a little bit bigger, we might get bigger micelles. Now if we make it even longer, we start now getting nanoparticles that are non-spherical. So these are cylindrical micelles, and this is a transmission electron microscope picture of these cylindrical micelles. And if we make it a little bit longer, we get something like polymersomes. So what all these particles have in common is that they're taken up by into the cells really quite efficiently by a process called endocytosis. So what we don't have to care anymore is about how hydrophobic the drug is that is on the inside. Now the pharmacokinetics is just given by the nanoparticle on the outside. So let's go back to our fructose micelles. And so we incubated now our fructose micelles either with healthy cell lines. So up here, there's some healthy cell lines, for example, like CHO cells or some macrophages. And down there, we incubated them with various breast cancer cells. And because these nanoparticles are now preferably taken up by the breast cancer cell lines, these cell lines, they start to light up because we labeled our nanoparticles with a little fluorophore. Once the nanoparticle goes inside, our cells become fluorescent. And this is a way now to measure the cellular uptake. And what we can see here is that we have a very good uptake by our breast cancer cell lines. So obviously, just attaching our fructose works really quite well, but the question is always, how many fructose molecules do we actually need? Is it enough having a few of them, or do we need them in a specific arrangement? Is this is sugar attaching enough? And the first example I would like to show you is where we made polymers with different lengths of our fructose polymer. So we had first 39 units, 89 units, and then 183 units. And all these molecule block copolymers give us micelles now. They have roughly similar sizes, but the bottom one has now a very, very hairy shell. So our polymers are in a very sort of hairy arrangement. And so what we find now is that we have a much, much better uptake by these longer polymers. And it doesn't matter what cell line we're using, longer polymers are much better in promoting this uptake. In order to understand it a little bit, we have to dive in a little bit deeper in understanding what the structure of these nanoparticles are. And the one way of doing that is, for example, neutron scattering or X-ray scattering. And what neutron or X-ray scattering gives us is, it tells us a little bit about, for example, how hydrated is the shell on the surface, how mobile are these polymers on the surface. And what we find here is that the longer the polymer is, the more mobile these polymers are. And by having a higher mobility of these polymers, we allow a better interaction with the lectins on the surface. So it basically means if we pack polymers very, very tightly, they're almost like a really tight toothbrush arrangement. They find it really hard to interact with these lectins. So we need something that is very mobile. So when we look at that, then we can probably say, well, if the mobility and the hydration is so important, then it is probably a good idea that we have a lower grafting density. Or to say with other words, does the grafting density play a role? And when I talk about grafting density, I talk about the way these polymers assemble so that they either have a very, very dense polymer layer or they have a very loose polymer layer. So our hypothesis is that this should perform better because we have better access to fructose. So what we are doing here is we are using one block copolymer, and now we process this block copolymer in different ways. And we are getting very different micelle particles with very different grafting densities. If we just look on the transmission electron microscopy picture, they all look the same. These particles are all around 30 nanometer in size. However, again, from sex measurements, we know that they have indeed very different grafting densities. So now again, we take these nanoparticles, we incubate them with cells. And what we find that we have indeed a much higher cellular uptake with the low grafting density. And we do that in various cell lines. It's always the same. So it almost feels that high grafting density, what we find here, led to reduced cellular uptake. And at this point in time, we can probably be pleased with ourselves because we said, well, our hypothesis is true. So if we have a bit more space, the binding with lectin is better. However, I would like to come back to this picture again here. I said the danger having something in a biological system is that it interacts with anything that is there. And it might interact with proteins you find in the blood as well. So what we always need to do when we work with nanoparticles, we need to understand how do these nanoparticles interact with particularly serum proteins. And so one way of doing that is that you take these nanoparticles and you do incubate them with either serum or you can incubate them with blood. And then you isolate these nanoparticles and you measure how much protein is on the surface. And what we find, there's really quite substantial amount of protein on the surface. So this means basically we started off with a nice sugar layer but now we have a layer of protein on the surface. So our targeting ability is reduced. So, however, what happens on top of it is not just that we have really a layer of proteins on the surface, but also this protein might now do something to these nanoparticles we don't expect. In that case, we found that our proteins managed to deform these nanoparticles, and we suddenly got spheroid particles. And that's really quite important to understand, because round particles have a very different cellular uptake pattern to spheroid particles. So what we really found is that when we have nanoparticles, we need to always consider that these nanoparticles might not be able to do their targeting jobs as we expected to do. So we started off with nanoparticles with fructose on the surface, expecting that these nanoparticles will interact with the cell membrane in a very specific manner. But what we found is that we actually have proteins on the surface that might really inhibit that specific uptake. So the surface of nanoparticle influences now non-specific protein absorption, and the amount and type of proteins that determine very much the fate of nanoparticles. So now we have these proteins here, and then suddenly you might find that your nanoparticles are taken up by the immune system very quickly. The other thing to consider is that proteins can do very interesting things to nanoparticles. They can deform them, they can disassemble them. So we always need to be careful. So, so far we just had nanoparticles, but we didn't have any drugs in there. And I said to you, we really want to use them to deliver drug molecules. So what happens to these nanoparticles when we deliver that, when we try to put in drugs? And there's in literature always a very common assumption that we start off with nanoparticles, and when we process them to put drugs in there, we don't have a change in particle size, and they always stay the same. And so there's always this concept of the Trojan horse. So it's this wooden horse, and we can't see from the outside what's really inside. And the other assumption we find in the literature that it's always better to put more drugs into nanoparticles. And it makes a lot of sense, because our nanoparticle is really just a carrier. It's like a mini nanoplastic bag. And really what we would like to deliver are the drugs on the inside. So the nanoparticles are really just waste products in the end your body needs to get rid of. So ideally, what we want is that our nanoparticles load the drugs nicely on the inside here, and our cell does not detect any drugs. So these nanoparticles interact very nicely with the cells and are taken up. So the question is now, or the hypothesis here is now, are more drugs better for drug delivery? And your first response probably would be, yes, it is, because the more drugs we have, the more toxic it is. So let's have a look at that. So here we have 2 nanoparticles, and we load them with platinum drugs. So platinum drugs are very commonly applied anti-cancer drugs. And in one nanoparticles, we put in a lot of drugs, and in the other particles, we put in very little drug. So now we would like to understand what the cellular uptake is. And here comes the first surprise, because if we have only a little bit of drug in there, then we find that we get a much better cellular uptake than when we have a lot of drug in there. So something is happening. Somehow our cells here, so up here we have some ovarian cancer cells, they somehow sense that there's a difference between these 2 nanoparticles. From the outside, they really should look the same. The only difference is that one has a lot of drugs, the other has little drugs. But the cells somehow knows that something is going up, is going on. So if we have more more loading, we have less uptake. And because we have less uptake, we also have less toxicity. So again, we need to understand a little bit more what's going on with our nanoparticles. In that case here, we did a lot of solid state NMR. And I don't want to bore you with details, but what we basically do in solid state NMR, we look at the chemical groups in there and we look at how they relate to each other. So for example, we look at the platinum drug and we have a look, how does the platinum drug, for example, relate to the glycopolymer, and how does it relate, for example, to other platinum drugs? And you can see already there's quite a difference here between the two. So what we learned from these this case is that if we have only a little bit of drugs in the core, we have a very high hydration levels of this whole nanoparticles. So it's swollen with water. And because it's swollen with water, it can interact much better specifically with the cells. We have a quite loose morphology, and it also allows for the drug to be released. However, if we have a lot of drugs here, the nanoparticle is very tightly packed, and the drug release is very much delayed. And also because it's very tightly packed, it's very dehydrated. So these chains on the outside can't really interact with our cells. So then in reality, a lot of effects the drug might display on the nanoparticle. In the simplest case, loading drugs might just change the size. Sometimes it changes the shape. We found occasionally that the drug doesn't stay in the core, it might diffuse into the shell. Or the drug simply affects the property of the shell. So, and of course, in the next step, this will influence how these nanoparticles absorb proteins. So some of them absorb less proteins than the other ones. So in the last 2 minutes, I just want to show you a little story which we just published recently. And I would like to go back to this picture here we had in the beginning about the inhibition of the invasion of viruses and bacteria. So bacteria infections are a very big problem in hospitals or in general. And it's really listed as one of the big challenges. In the current treatment of bacterial infection are, of course, antibiotics, but we developed a lot of antibiotic resistance recently. So the solution is, of course, to either look for new antibiotics. People look for alternative approaches, for example, antimicrobial peptides. And these are peptides that, for example, make holes into bacteria and make them inactive. But the other approach is that we simply inhibit the invasion of bacteria. So we have molecules in here that stop these bacteria from attaching to our cells. And very often these nanoparticles are based on sugars like mannose, for example. So the problem is just by using these sugars like mannose is that there's an abundance of mannose receptors throughout the body. For example, the liver has a lot of mannose receptors. So it's not really terribly specific. However, there's one sugar that's really quite specific to the bacterial world, and that's trehalose. So trehalose is a non-reducing disaccharide made from two glucose molecules bound together. And the interesting is that trehalose is widely used by bacteria in other cells, but we don't have any biosynthesis of trehalose in mammals. And bacteria use trehalose, for example, as a source of energy, as a defense mechanism, or also to invade other cells. So the idea is now that we use a polymer based on trehalose to inhibit the invasion of bacteria. And so what we made is this trehalose polymer, but we also made two controls. And one control is a glucose attached on the 6th position and a glucose attached on the 1 position. Then we took these polymers and we attached them to gold nanoparticles. And then we studied the inhibition assay. So if we incubate cells, healthy cells with bacteria, the bacteria attach to the cells. However, if our material is successful, it should attach to the bacteria. And as a result, our bacteria cannot invade the cells anymore. And in reality, this actually works quite well. So if we just take our normal glucose, we get a slight inhibition of an infection of Staphylococcus aureus to HUVEC cells. However, if we have now another glucose, we even reduce it further, and we have a further reduction with trehalose. So this is not very obvious here, but we know that trehalose is really quite efficient in inhibiting the infection. And the reason is because these trehalose-based gold nanoparticles, they attach now here to these bacteria. And by attaching to the bacteria, it stops inhibition. And when we count these gold nanoparticles on these bacteria, we found that the trehalose attaches very well to these gold nanoparticles. So the inhibition here, there might not really be as efficient as maybe other particles, but one thing to consider that is trehalose is nanoparticles. They're non-toxic, so they're not dangerous to any other cells, and they're also very specific to certain bacteria. So that brings me to my conclusion. So I hope I could show you that glycopolymers are in fact sweet. They're not just sweet because they taste sweet, but they have a lot of excellent applications. They can, for example, assist in the uptake of nanoparticles by cancer cells. However, we need to understand how these sugars are presented on the surface of these nanoparticles. And the binding can be very much influenced by the length of the polymer, how the polymers are arranged on the surface. But we always need to be careful. A biological system is something very, very complicated. And we always might have interaction with other cells. We might have interaction with blood proteins. And so with that, I would like to thank funding from the Australian Research Council. And of course, I would like to thank the students who've been doing all this work. And I would like to thank you for your attention.
Great. Thanks a lot, Martina. We have a couple of questions here. So because of the time constraints, we're probably going to go with two and see if we may have, we may be able to squeeze in a little more. So the first one is from Dr. Arain, and he wonders if you could use copper catalyzed azide alkyne click reaction to synthesize thermal responsive glycopolymers for the inhibition of ricin. And if that's a possibility, what is the effect of spacer between polymer backbone and lactose?
Yeah, it's the same that if we have more mobility of our sugars, we usually can enhance the interaction. So if we have a longer spacer, we do get better mobility and therefore better binding. So yes, the question is flexibility is always a good thing.
All right, thanks, Martina. Next question is from Kelly Bologna, and I think she wonders, what about the reversibility of bacteria inhibition through trehalose or other means? Is this necessary or is that feasible? I will send that to you too.
Okay, what about bacteria inhibition? Well, it's probably not necessary. I mean, if you can attach these nanoparticles on the bacteria, we probably can clear them easier from the body. So we haven't really looked at the reversibility yet. It's a very good point, actually. The question would be, can we sort of flush these nanoparticles off the surface and how long would they stay there? So no, we haven't looked into that. It's a good question. Yes, thank you, Kelly.
Okay, thanks, Martina. And I think we could have one last question. It's about how are the nanoparticles metabolized after they enter the body? I mean, that.
They're usually cleared after a while. So we did biodistribution studies on some of them, and we find, of course, ideally we would like them to go to the tumor, but a lot of them go to the liver. They stay in the liver, and then they're cleared from the liver again for the simple reason. In most cases, we use a block copolymer system, and that block copolymer system can fall apart into single polymers again, and they are cleared from the liver. So they don't accumulate for a very long time. Having said that, we did a study on other mice cells where we stabilized it, and these nanoparticles stayed in the liver for quite some time. So this is not something we want. We want to have clearance again, but they're usually cleared. They stay in the liver for a few days, and then they're gone.
Okay, thanks again, Martina, for your presentation and your answers for the questions. We still have a couple more, but we are really running over time. So perhaps these audience can get in touch with Martina after this using email or other means. Next, we will move to our fourth talk, and also the last talk for today from Professor Zhang at Tsinghua University in Beijing, China. So Qiang graduated from the same university in 2004 with his BS degree, and then he also obtained his PhD from Tsinghua in 2009. He then held research associate postdoc research fellow positions at his Western and also the Fritz Haber Institute of the Max Planck Society in Germany. Now he is a professor at the engineering department of Tsinghua University. He held the Newton Advanced Fellowship from Royal Society in the UK and the National Science Fund for Distinguished Young Scholars. He was awarded the Tianzhao Award from the International Society of Electrochemistry, Science and Technology Award for Chinese Youth, and first prize of Natural Science Ministry of Education. And today, Qiang is going to tell us about emerging energy chemistry at lithium metal and electrolyte interfaces in working batteries. I will now pass it to you, Zhang.
Okay, thank you. Thank you, Xin, for your kind introduction. This is Qiang from Tsinghua University of Beijing. It's my great honor to join the Angewandte Chemie Fall Symposium 2021. I will share my recent research on emerging energy chemistry of lithium metal anode and electrolyte interface for safe batteries. So first, I will introduce the background of this field, and then I will move to the fundamental of energy chemistry for lithium metal anodes, and then I will show how to design a good electrode material for rechargeable batteries. You know, from the view of blue energy consumption with industrialization, we consume more and more coal and oil worldwide, and the energy, the carbon dioxide emission was rising quickly. This, if we check the warm gases emission, we found the carbon dioxide was the dominant one, while the methane and nitric oxide was also contribute to the global warming. And then carbon neutralization was strongly considered worldwide. In China, our president showed the peaking carbon dioxide emission by 2030, and we are doing great effort to reaching carbon neutrality before 2060. So it is quite challenging goal, and how to reach that. So if we check carefully about the energy consumption, it is found that electricity and heat production occupy half carbon dioxide emission, while another 30% was contributed by the industry, and 10% was contributed by transportation, and 10 was another parts. So how, is it possible to harvest the electricity without carbon dioxide emission? The answer is definitely yes. The reason is that there is some emerging technology like the solar energy and also the electricity harvested from wind. However, they can't play the dominant role before 2030. If we check the amounts of energy harvest, it is found that coal and oil was the dominant one before the carbon peak. Well, the ratio of renewable energy will be around 70 if we reach the carbon neutralization. So in this case, how to realize this goal? Because you know the chemistry was shifted from carbon to lithium metal from one viewpoint. The reason is that once we harvest energy from solar, from wind, they are no continuous one. You know the sun will can get electricity in daytime, but in night we have to get energy from the storage device. And also, you know, in summer the sun is very strong. It's always sunny. While in the winter the sun is not very strong. So there are also long-term energy storage requests for the renewable energy. So in this case, the energy storage not only match with the gas electricity production, it's also strongly related from the electric grids as well as the final energy consumption. So based on this consideration, the lithium-ion battery was strongly considered recently. The success was lying in the contribution by these three scientists. Professor Stanley Whittingham proposed the concept of lithium-ion battery about 50 years ago, and they improved step by step. And the current one, the lithium cobalt oxide paired with carbon anodes, was widely accepted. And this introduced a lot of new devices like cell phone, like laptops and electric vehicle worldwide. So in this case, if we consider the request of future energy storage as well as the current request for electric vehicle, it is found that the safety, the high energy density was strongly requested. Here I show the rising demand for batteries. It is found that the electric vehicle was dominant in the coming 10 years, while the energy storage was still rising quite quickly. So based on this consideration, is it possible to propose better energy storage device based on electrochemistry? If we track the history of the battery, it is found that energy density increase is rather slow. If we check the lead acid battery or nickel cadmium battery, it is found that energy density rising quite slow. The emerging of lithium-ion battery based on emerging lithium and chemistry, it is found that it's rising quickly. But however, if the battery based on carbon anodes, the energy density was always below 350. Well, if we try to develop some application like unmanned vehicles or other requests, which request the battery with very high energy density, it is found that the emerging new energy chemistry was requested. So with this consideration, we try to plot the cathode versus anode with different system. And it is found that the carbon replaced by silica, the energy density, whether gravimetric or volumetric, will be increased gradually. If the anode was fully replaced by lithium metal anode, the high energy density was always required. However, there are several challenges. The first one is how to utilization lithium metal in high utilization. In fact, if we check the electro cathode and anode pairs, it is found that how to reach the high utilization was the first step. And second one is that in a working battery, during the repeated plating stripping, the lithium metal anode was not always deposited in continuous form. Some dendrites will be achieved on the surface, and the dendrites can penetrate through the separator and they introduce safety issue. While the high active lithium metal anode will touch the electrolyte and they introduce some side reaction, which introduce a lot of gas and some solid byproducts. In fact, the gas products include hydrogen, methane, ethylene. They are quite dangerous. So in this case, if we purchase a high energy dense battery, it is found the chemistry happen at the interface between these metal anodes and the electrolyte was quite challenging. So we try to focus on the interface and try to probe what's happening here. In fact, if you try to drop this metal anode on surface, there always some chemical absorption on surface. Inside, we call this inner homogeneous plane, in which some ions prefer to absorb on the surface. While in the outer homogeneous plane, there are some ions, and also there are long diffusion region in which the ion will diffuse from the main phase to the interface. So in this case, what's happened in the interface and how to regulate the interfacial reaction? So based on this consideration, we try to probe the electric double layer at the interface, and we try to understand what's happening here. In fact, there are always solid subproducts on the surface. The solid products constitute of many oxide fluoride carbonates and also some organic lithium salts on the surface. They prefer to be a film on the surface. They will expand or shrink based on the plating stripping of the lithium metal anodes. In fact, if you check the composition of the anode ICI carefully, it is found that the composition and its structure is highly depends on the degree of discharge on the charge of the battery. So there are some emerging characterization methods that are present in this figure. It is found that a lot of advanced characterization can be employed here. For example, we can use some electron or X-ray beam or some light to touch the surface, and then we can get the feedback about the composition, about the bonding, about the chemical composition of the surface. We also can sparking the surface and get the depth profiles. And also there are several molecular modeling methods which can provide the information about the electron structure, while the electrochemical technique can prove the ion conductivity and electroconductivity of the working interface. So in this case, it is possible to carefully describe what's happened between the electrolytes and the metal anodes. And in this case, we try to probe not only from the viewpoint of the thermodynamic control, we also can to probe what happened from the viewpoints of kinetic control and try to control the interface structure. So in this case, we try to answer what's the origin of interfacial side reaction? What's the side reaction happen there? And also, there are a lot of solid products deposited on surface. So what's the solid products and can we regulate the composition and its distribution? So first, we try to probe what's happened at the interface. The lithium metal anodes or the BBC crystal and is not, and we try to combine them with some carbon host. Well, from the viewpoint of the electrolyte, it is found they are quite complex. The lithium in the solvent was not individually paired. It can be coordinated with the solvents and form unsolvent complex structure. Once the lithium ion coordinates with the solvents, it is found that the lumen of the solvents can be reduced significantly. So in this case, it's preferred to attract electron, while the exposed alkaline metal can provide electron directly to the unsolvent complex. So with this consideration, we try to probe what's happened once lithium ion take one oxygen and then electron transfer to there. It's found that the decomposition of the organic solvents was quite easy. And this step was with quite few energy barrier. And then the lithium ion prefer to attack another oxygen. And once the electron reach that site, then the ethylene gas can be available in the electrochemical system while a lot of solid products can be deposited on the surface. In construct of lithium-ion solid complex rules, the direct decomposition was always with a high energy barrier. So in fact, we try to compare the energy barrier with the lithium-ion solvents and the direct decomposition. And they found that the ion-solvent complex provide a quite easy and facile routes to help the electrolyte decomposition. So in this case, if we want to stabilize the lithium metal anodes, we have to think how to stop the electron transfer from lithium metal anode to the ion-solvent complex, and how to retard the interfacial reaction, because from the simulation, it is found that the formation of ethylene was happened around 600 femseconds. It's a quite fast process, and the only way we can modulate is to reject the electron transfer from fresh metal to the electrolyte. So in this case, we try to observe, is it possible to coating the lithium metal anode with a film to protect the surface? In fact, if we try to dip the lithium metal anode to the pure organic solvents, there is quite few gas evolution. While in the salts containing electrolytes, the gas bubble formation was quite quickly. And such phenomenon was a universal one, because in pure solvents, whether in DOL or DME system, the gas evolution was quite slow. And we also try to compare with other electrolytes, and the results was quite similar. We found the importance of the ion-solvent complex structure was quite important for the decomposition. So in this case, we use ab initio calculation to prove the bonding energy. And it is found that in the lithium-ion solvent was quite stable and with strong bonding in EC or DME electrolytes, in which the ion-solvent complex bonding energy was higher than the salt bonding. So in this case, it is found that in some certain system, the lithium salts prefer to decompose into titans and ions, while the different ions have different bonding energy with lithium salts, for lithium ions. For example, the nitrite ions always prefer to have a strong bonding, so it's preferred to interlink with the lithium ion in working electrolytes. Based on this consideration, we try to characterize the solid products on the surface. So here is the low concentration electrolytes induced SEI. This one was the high concentration electrolyte induced SEI. And it found that with increasing of the salt concentration, the inorganic compounds was increased significantly. The contribution of lithium fluoride, lithium carbonates, and lithium oxide was increased significantly. And this is also easily confirmed by the TM observation, in which a lot of inorganic crystals can be found on the surface, while for the SEI in the low concentration electrolyte, it is found that many amorphous structure on the surface. So this indicating the ion-solvent complex structure can regulate the SEI composition. And we also try to probe, if we change the electrolyte structure, then what's happened? For example, in routine carbonate electrolytes, the capacity versus potential was quite rapid decay. Well, for the case of high concentration electrolytes, it is found there a small peak at the beginning, then to the platform. Well, for the case of low colloids, high concentration carbonate electrolyte, in which we try to introduce some solvents, they don't dissolve salts, but they can mix with another solvent easily. So in this case, the peak reduced a bit, but there's still a peak before the platform coming. Well, the final electrolyte was weakly solventation electrolytes, in which it have a very low dielectric constant. The electrostatic interaction will be very strong, and the ion prefer to be coupled in the electrolytes. There is also a wide peak in the surface. So we try to assign this is the decomposition of the solvent and salt for the ICF formation. And then we try to fitting what's the evolution and the growth behavior of the inorganic SEI. It is found that at the beginning the slope to 1, but the coming was to 3, indicating the progressive nucleation on this metal at the beginning, and then as a 2D dimensional growth in which the ICI attached on the surface, and finally they overlap with each other to get a continuous film to reject the direct contact of these metal anodes with organic electrolytes. So in this case, we try to understand what's happened here, and then how to regulate the lithium-ion solvent complex structure. Then we can promoting the lithium-ion reduction, and also we can resistance. We can try to build a stable SEI to avoid decomposition. For instance, we try to introduce fluoride ethylene carbonates, which are very widely accepted film formation additive in most batteries. It is found that the FEC molecule prefer to in the shell structure of lithium ion, and such structure reach to lithium metal anodes. Once the lithium metal was exposed, the lithium ion solvent complex will decompose, while the FEC will provide more lithium fluoride deposition on the surface. And from the observation, it is found the lithium metal prefer to uniform growth, no dangerous formation in routine carbonate electrolytes. So this reason was lie in the stable interface between lithium fluoride and lithium metal. With the addition of FEC, we found the lithium can contact with FEC easily, and this helps the binding of FEC with more FEC molecule in the shells. And once we try to check the fluoride products on the surface, whether by fluoride or by lithium peak by photonic spectrum, it is found that the lithium fluoride was sharply increased with the addition of 5% of FEC. So in this case, the introduction of FEC provide a uniform and high ion conductive but reject electron transfer layer of ICI, and this guarantees the Coulombic efficiency of the lithium metal anodes was quite high, and the polarization on the interface was small and relatively stable. So this is quite important for working cell in which it's rendering the full cell of lithium metal pairs with NCM, nickel, cobalt, magnesium oxide with uniform and with stable cycling. So in this case, it is found that the electrolyte additive can regulate the lithium ion chain structure, and this helps the stable ICI formation. And in fact, if we try to analyze the reaction, it is happened between lithium metal anode and ion complex. So if we try to introduce the electrolyte with different ions, for example, we mentioned before, nitrogen ions prefer to attach with the lithium ion. So in this case, once we introduce nitrate ions in the electrolyte, it is found that the peak shifts of NMR in the oxygen peak, can observe the peak shifts. And we also get the distribution of the lithium ion, lithium oxygen, lithium nitride, and lithium fluoride coordination. And it is found that these items prefer to interlink with lithium easily. And once the ion complex receives electrons, then it helps the composition of the ions quite easily. This will introduce a lot of nitride oxide products on the lithium metal anodes. Also, lithium fluoride can be observed in large amounts. And this rendering the lithium metal anodes with an inorganic compound dominant surface, which rendering the cell with quite large exchange current density, and this is quite important to help lithium ion diffusion in a working cell. The large exchange current density help the battery with high rate performance. This also can be observed by the electrochemical test results, in which the lithium nitride addition, the cell with more electrolyte decomposition, and if we try to record the working window, it is found that can be enlarged from 3.5 to 4.5. So this helps the electrolyte match not only with sulfur cathode, the nickel magnesium cobalt oxide system can be also applied in this case. And once we try to compare the second life, it is found the regulation of the unsolvent complex structure can regulate the protection of the ICI, and this rendering the cell with quite long second life as well as stable polarization in the whole life. So this is quite important to provide the feasibility of cycling the cell with a long second life. And also, I want to try to introduce how to weaken the lithium and salt interaction, because if the lithium can coordinate with salt strongly, the salt can decompose fully. Well, if we try to reduce the dielectric constant, then the electrostatic interaction will be reduced, and then the lithium salts dissolved, but it appears not in the lithium ion in dominant states. The reduction of dielectric for the strong electrostatic interaction. And in this case, if we try to observe the states of lithium ion, it is found the pairs and also the aggregates of lithium salts can be dominant with the introduction of dielectric solvents. And in this case, the lithium ion was not dominant move in the unsolvent complex structure. The couples or the aggregates was the dominant one. And it is found that such case of electrolytes can rendering the cell with fast charging property, because the lithium pair move quickly than lithium ion solvent complex in working electrolytes. So in this case, we try to regulate the interfacial reaction to protect lithium metal anode by insert achieved ICI. And also, if we want to build a full cell, we have to admit that the 30 micrometer lithium metal anode was too large volume shrink or expansion for working battery. So in this case, we try to introduce the addition of host, in which it is found that host not only provides space for volume expansion or shrink, we also provide the lithium philic sites in which they prefer to unking the lithium ion by lithium bonds, and the strong binding of the cell with strong binding energy. And this helps the lithium nuclearization was in small size, in high density, and this indeed helpful for lithium metal anode deposition into uniform film, not lithium dendrites in a working cell. So the lithium bond theory was quite important and necessary to guide the lithium metal anode to in uniform deposition. And in the active material system, once we try to dope the carbon with heteroatoms, it is found that there are always several defects. The defects not always bad. It is found that some defects like stone walls interact structure can also provide lithium bonds to the lithium ions. So in this case, it also benefits for uniform deposition. And finally, we try to integrate the least cone cell into punch cell. It is found the case is quite different, because in cone cell, the aim is try to probe the chemistry and also try to demonstrate the feasibility of the engine material. Well, in the case of full cell, we try to actively build a high energy density battery. So in this case, we firstly probe what happened in working cell. We try to probe the charging discharging behavior of this punch cell, and it's found that once the capacity and the current density increase gradually, the interface is not stable, and there are a lot of evolution in the structure of the lithium metal anodes. Well, if we try to apply a too large current density, if the short circuit was directly observed, which also confirmed by the observation, there are a lot of lithium metal residues, but the lithium dendrites penetrates through the separator and induce the cell diet. And in the mild condition in which the lithium metal anode become powder, the dead lithium was dominates and the polarization increased significantly. So in this case, how to overcome the scale-up effects that corresponding to the strategy develop or propose at the mild condition, how to scale up into the harsh condition. This still require many chemistry and chemical engineering investigation, which call for not only the mechanism of the transport phenomena and individual reaction, we also call for engine material to realize that. And in fact, we try to use calendar process to build composite lithium metal anodes, and the direct calendar provide large amounts of lithium metal anodes, in which it provides the feasibility to demonstrate the chemistry in a full cell. And it is found that the feasibility of 6 watt-hour per kilogram can be available if we can impair the some high voltage potential anode and cathode. And finally, I'd like to focus on the safety issue of the lithium metal batteries. As you know, the lithium metal match with moisture is there always hydrogen erosion. Well, in fact, in Biosynthesis, we are working on lithium-ion battery with plating more lithium metal. There are always a lot of lithium metal deposition once the 100% of lithium metal deposition. It is found that, for example, by 25% or overcharge, it is found lithium can be still uniform deposit on the surface, while for the 50%, some lithium penetrate deposits on among particles. And we also test the overcharge and also new penetration test. It is found that once the uniform lithium plating, the temperature rise was not too large. Well, for the lithium dendrites formation, the temperature or the tips and the edge will be very high. And also for the new penetration test, the temperature rise was very large. So based on this consideration, we found the importance of lithium metal uniform deposition, in which we try to regulate lithium metal deposition by the electrolytes. It is found that the uniform deposition of lithium metal anodes was necessary step. And also we found the addition of 25% of overcharge is still safe in most cases, especially once we guarantee the Coulombic efficiency was more than 99.5%. The superior safety performance can be available in this case. So this provides the fundamental for the addition of lithium at the initial stage, which can help the lithium metal anodes with a long cycle life. So finally, we have some review on this topic. We try to summarize the fundamental interaction and also lithium bonds, as well as how to get safe lithium metal anodes. So based on my understanding, the fundamental of the interface between lithium metal anode and electrolyte was quite important. And there are several Fourier mechanisms which should pay attention to the SEM formation and how to guide the lithium metal anode uniform deposition in a 3-dimensional host. And recently we found the introduction of new and emerging research methods like machine learning and artificial intelligence was quite important. They are quite helpful to probe and predict the cycle life of the whole battery. So in contrast of previous research, based on to my understanding, the reason of lithium metal anodes have a lot of new coating film. That's quite different from before. And the addition of the composite host, this provide emerging energy chemistry to guide the lithium metal deposit to the right size. So this is quite important to get this metal anode deficit in a safe and reliable way. And the coming of lithium chemistry in contrast of carbon chemistry was quite important. So finally, I'd like to thank the editor office of Angewandte Chemie, and also I'd like to thank the founding and also Macro member for their great job to provide opportunity to share my results. And I'd like to answer any questions from all of you. Thank you very much.
Great, thanks a lot, Zhang, for this excellent presentation. Great, so I don't see any question from the Q&A box, so I will just have one last, oh, here we go. Okay, so we have one from Gamini about how to control metal dendrite formation. I think that's that's a quite
that's a good question, and it's very important for the utilization of this metal anode. The first one is that try to build a stable SEI by regulation of the reaction happens between electrolyte and this metal anode. And the second one is that help this metal nucleation in high density and they working at a small localized current density. So this helps this metal get in mild condition, not harsh condition. So the combination of ICAI and host was a practical method to stop the dendrites formation in a working battery.
Great, thanks, Zhang, for your answer. And I see one more question here. Okay, oh, actually no question. Okay, I would like to take this opportunity to thank all four advisor editors giving wonderful presentations for today. And for all those who have registered for the Meet the Editor session, you can find the link to the session in the chat box, and we will see you there in 5 minutes. Thanks again.